Good morning dear students and welcome to Genesis. Well, this video of mine is in continuation to my lectures on the chapter Respiration in Plants. Last ke video mein we have already understood how experimentally we can prove that at the time of germination seeds require oxygen. Now in today's lecture we will prove that carbon dioxide gas is liberated by germinating seeds. Now for this we have taken two round bottom flasks, and in flask A we have kept seeds which are soaked in water and we have placed them over moist cotton. So this moist cotton will provide enough moisture for the seeds to germinate. Whereas in set B we have taken boiled seeds, and you already know that boiled seeds are basically dead, and we also make sure that we wash them in antiseptics because we do not want any bacterial growth on these dead boiled seeds, because these bacteria then will perform respiration and can give out carbon dioxide which can impact the result of the experiment. So set B is a control of this entire experimental setup. Now we are going to close the mouth with cork and we are going to observe. What we are going to observe is that what kind of gases are produced, especially if carbon dioxide gas is produced during germination or not. Prove that we remove the cork from the mouth of both the flasks and we tilt the flask, bringing it very close to a test tube. But this test tube is just not general test tube. You have kept lime water in the test tube, and we know that when carbon dioxide mixes with lime water, it turns lime water murky. So it's an absolute test of carbon dioxide. So we open the mouth of the flask and we tilt it over the test tube and we try to observe what happens. Now in case of situation A, when we do it with this round bottom flask A, we observe that this lime water turns milky. This proves that there is enough carbon dioxide gas that must have been produced within the flask. And who must have produced it? It must have been produced by these germinating seeds, because they got ambient conditions and they utilized oxygen and released carbon dioxide during the process of germination. And this carbon dioxide being heavier, it comes down from the flask and mixes with the lime water and thereby turning it milky. Lekin when we do this same experiment with round bottom flask B, हमें कोई भी change observe नहीं होता है. Reason being, यहाँ जो seeds हैं, they are anyways dead, so they are not germinating, and since they are not germinating, they are not going to release carbon dioxide. Hence, it is proved that during the process of germination, seeds oxygen consume जैसे करते हैं, वैसे ही they release carbon dioxide. लेकिन इस पूरे experiment के दौरान, this was very evident that this part is quite tricky, because you really cannot see carbon dioxide gas coming out from the round bottom flask into the test tube, right? So we need a more concrete experiment. So the next experiment that we are going to do is a more concrete experiment to prove that CO2 is released during germination. Now in the second experiment we have taken 4 conical flasks. Okay, this is flask A, B, C and D. But in flask A we have kept a solution which is potassium hydroxide. See, property of this chemical is it absorbs carbon dioxide. In flask B we have kept lime water. In flask C we have kept germinating seed. And in flask D, again we have kept lime water. There is a reason for this sequence. So the first one is KOH, then we have lime water, germinating seed, again lime water. What we are trying to do is, we have connected all these 4 conical flasks with the help of bent tubes. You can very well see they are connected. So now with the first tube, we allow air to enter. So when air enters into the conical flask, it will have carbon dioxide into it. Now what we make sure is, this potassium hydroxide will absorb all the carbon dioxide that is present in the air. Now from here the air passes to the next conical flask, but now we know that there will be no carbon dioxide in this air because all of it has been absorbed in flask A. So now in this lime water, what is expected is there will be no change, and that is what happens. There is no change in the lime water because there is no carbon dioxide in the air. Now from here the air goes to the flask C, which contains germinating seed. Now these germinating seeds require oxygen, so they will take oxygen from the air, and in return during the process of respiration they are going to produce carbon dioxide. Now again the air that comes out from this flask will contain carbon dioxide, and as it enters into flask D, the lime water now turns murky. The reason is, here there is carbon dioxide in the air, and 100% of the air has come from flask C. Then it must have been produced in flask C, because before that there was no carbon dioxide in the air. So hence proved that germinating seeds at the time of respiration produce carbon dioxide. This is another experiment just to prove that during respiration CO2 is produced, but now we are not taking germinating seeds. We have taken one whole plant. The plant that we have taken is germanium, and the setup is almost very similar to the last experiment. We keep two conical flowers on either side of the plant and they contain lime water. But remember, the pipelines which are entering into or coming out of it, they are like connecting the three jars together. So it is flask A, this is flask B, and this is the potted plant which is covered by a bell jar. And we also make sure that the ends of the bell jar, they are sealed with Vaseline, because we do not want any air coming in or coming out of the experimental setup. There's one more catch here. As the air enters into the flask A, it has to go through lime water first, and lime water has same property as potassium hydroxide. It absorbs carbon dioxide that is present in the air. So we can make this comment that the air which is actually entering into the flask A is without any carbon dioxide, because all of it has been absorbed by soda lime. And that is what happens, because as the air enters, we find there is no change in the lime water. That means the lime water does not turn murky, hence prove that the air has no carbon dioxide. Now the same air from flask A will now enter into the bell jar containing the plant, and after some time the air from the bell jar will now enter into the lime water containing flask, will enter into flask B that also contains lime water. Now what basically happens here is, though the air which comes inside has no carbon dioxide, but it has oxygen, and what the plant requires for the process of respiration is it is going to consume oxygen and liberate carbon dioxide. So there is liberation of carbon dioxide by the plant. This carbon dioxide will travel through this pipe and will enter flask B. Flask B contains lime water, and hence it will change the lime water milky. Proved that carbon dioxide gas must have been produced by the plant, because there is no other medium through which carbon dioxide can enter into the flask. Now remember one thing, here you are not working with germinating seed but a whole grown plant. So plants perform respiration and photosynthesis together, and in presence of sunlight, when they perform respiration, whatever carbon dioxide they produce is utilized by the plant during the process of photosynthesis to produce food. So if you perform this experiment out in the sun, you're not going to see the result. So the way to perform this is either you keep the experimental setup in dark, or you cover the bell jar with a black cloth. So that will ensure that the plant will not perform photosynthesis and will not utilize carbon dioxide, and that carbon dioxide can be utilized to change the lime water milky. Now we will prove another property of germination, that during germination the seeds produce heat. So for this we have taken two thermoflasks. Thermoflask, you know, these are the containers which do not allow heat to pass out. It remains trapped within the flask. Okay, so we have taken two flasks, flask A and B. In the flask A we have taken soaked germinating seeds, and in flask B we have taken dead seeds, and you already know what we have to do with it. We wash it in antiseptic so that there is no bacterial growth. You clog the mouth of the thermoflask and insert thermometer inside it and observe what's going to happen. After some time we will find that the reading of the thermometer of flask A, which contains germinating seeds, increases in comparison to the flask B, because in flask B there is no change in the reading of the thermometer. This clearly shows that only germinating seeds during the process of respiration, they produce heat, which we do not find in flask B. So with this I finish today's class. If you have liked my teaching, subscribe to my channel, share it with your friends, and do not forget to post your doubt. And yes, do not forget to revise. Thank you.